Hello everyone, I am Arindam Pal. Today I will be talking about a topic transcription which is quite important from the perspective of NEET examination. Now, in the NEET exam, this particular topic is coming under the chapter Molecular Basis of Inheritance, and the expected weightage of this particular chapter in the NEET exam is around 8% to 12.5%. And as we are seeing that year by year, the number of questions are going to be asked from this particular chapter is continuously increased. So that's what the importance of this particular chapter is. What is transcription? Transcription is basically a process by which RNA or the ribonucleic acid is going to be made within a living cell from a DNA template. In the eukaryotic cell as well as in the prokaryotic cell, transcription is a common and fundamental machinery. Now, why this transcription is necessary? Transcription is needed to produce the RNA which is eventually going to translate into protein, and we need protein to sustain the life. So to produce protein to sustain the life, we need RNA, and that is why we need the process called as transcription. Now, transcription is eventually happening from gene level. What is gene? Gene is basically a portion of DNA that is going to produce one particular type of protein, or in prokaryotic cell it is going to produce more than one type of protein. So this is what the construct of a gene is. Gene is basically containing three portions: promoter, coding region, and terminator. Promoter is basically that particular region of the DNA where during transcription a polymerase is going to bind. This is basically a DNA dependent RNA polymerase that is going to bind in the promoter region, and binding in the promoter region, the RNA polymerase is going to pass through, it is going to move through the RNA coding sequence of the gene. And in the terminator region, there is a termination sequence or the stop sequence, and there the polymerase is going to dissociate from the gene, and that's how the gene is going to be transcribed into RNA. Now, what is cistron? Cistron is basically the coding region of this particular gene. Now, this cistron could be of two types: polycistron or monocistron. In prokaryotic cell, what happens, under one promoter, more than one gene construct is actually there. So that is why they can produce more than one protein upon binding of polymerase in only one promoter. But in case of eukaryotic cell, each of the genetic construct is having their own promoter. So that is why they are monocistronic. So in eukaryotic cell, there are monocistronic genes, but in prokaryotic cell, there are polycistronic genes. Now, what is needed for transcription machinery? There are some basic requirements for transcription. One is template DNA, DNA dependent RNA polymerase, transcription factors, ATP for energy requirement, and ribonucleotides, which are the building block of RNA. We will look into each of them. Template DNA. What is template DNA? Template DNA is basically a stretch of DNA based on which an RNA is going to be synthesized. Now, RNA polymerase will actually bind to one particular portion of the DNA, and it will move through the DNA from its 3' to 5' direction. And this is how the RNA polymerase will actually turn into a productive enzyme to produce RNA. So this is what the necessity of template DNA. What is RNA polymerase? RNA polymerase is basically the engine of the transcription machinery. That is basically a DNA dependent RNA polymerase, because it is taking the DNA as a template strand. That is why it is DNA dependent. It is RNA polymerase because it is going to polymerize the ribonucleotides into RNA. So that is why it is called as RNA polymerase. With respect to RNA, the ongoing newborn mRNA, it is going to move, the polymerase is going to move from 5' to 3' direction. And it could be of three types mostly, but there are other type of RNA polymerases as well. Three main type of polymerases are RNA polymerase 1, 2, and 3. And this table is very important from the perspective of NEET exam. RNA polymerase 1 is necessary to produce rRNA, that is the ribosomal RNA. RNA polymerase 2 is needed to produce mRNA, the messenger RNA, and the heteronuclear RNA called as hnRNA. And RNA polymerase 3 is needed to produce tRNA, 5' RNA, and the small nucleolar RNA or the snRNA. What is transcription factor? It is another requirement of the transcription machinery. Transcription factors are the proteins apart from the polymerase enzyme. Apart from polymerase enzyme, we need some of the proteins to continue the transcription machinery. Three type of transcription factors could be there. One is needed to start the transcription machinery, called as initiation factor. One is there to continue the transcription machinery, called as elongation factor, which are mostly found in the eukaryotic cells. And there are some termination factor which are needed to stop the termination process. We will look into each of them in the consecutive slides. What are the steps of transcription? How this transcription machinery is actually going to happen? There are three steps to complete the transcription process: initiation, elongation, and termination. What is initiation? Initiation means starting of transcription. In the starting of transcription, RNA polymerase and the sigma factor, that is an initiation factor, going to bind with the template DNA, and it is going to form a bubble-like structure called as replication bubble. Now, how this particular RNA polymerase is going to bind the particular region of the DNA template? It is always going to bind with the promoter region of the DNA template, as we have discussed before. And the promoter region is going to have a particular conserved sequence called as TATAAT, or betterly known as Tata box in the eukaryotes and archaea, and the same sequence is called as Pribnow box in case of prokaryotic cells. So in initiation, what is going to happen? The machinery, the transcription machinery, or the RNA polymerase enzymatic machinery is going to bind with the DNA template, and it is going to start the process of transcription. In elongation, what is going to happen? The sigma factor is going to dissociate from RNA polymerase, because the sigma factor is going to block the tunnel from which the ongrowing mRNA is going to come out. So if the elongation process is happening, that has to occur with the dissociation of sigma factor, because if sigma factor is not going to dissociate, the machinery will not be able to pass through the construct of the gene. So this is called as elongation process. In the termination process, there are two type of termination process. One is hairpin loop based termination, one is rho factor dependent termination. In the hairpin loop based termination, the mRNA is going to produce a hairpin loop, and that will eventually halt the RNA polymerase enzyme, and RNA polymerase will actually dissociate from the template DNA, and that's how the termination will occur. That is called as rho factor independent termination, because no rho protein or termination factor is going to be involved in this particular termination process. Another type of termination is rho factor dependent termination process. In this particular process, what actually happens, in this process also hairpin loop is going to be formed, but rho factor is a termination factor which is going to bind with the mRNA, and it will actually cleave the hydrogen bonds between the mRNA and the template DNA. So that's how the mRNA will actually detach from the template DNA strand. So that is another type of termination called as rho factor dependent termination process. So this is how the mRNA is going to be produced, and it will come to the cytoplasm to produce the proteins. Some more steps of transcription: capping, splicing, and tailing. So what actually happens in the eukaryotic cell, we have some introns which are called as the non-coding region of the gene. So we need to splice out these introns from the mRNA to produce protein. Now the steps are consecutive: capping, splicing, and tailing. In capping, what actually happens, a methylated guanine residue is going to attach at the 5' end of the heteronuclear RNA or newly formed pre-mRNA. This is called as capping. In the next step, what actually happens, introns are going to be spliced out by forming spliceosomes. Spliceosome is basically a protein complex that is necessary to cut out the intron regions from the pre-mRNA, And then at the 3' end of the pre-mRNA, polyadenylation is going to occur. Polyadenylation means adenine residues are going to be added at the 3' residues. So that's how a mature mRNA is going to be produced inside the nucleus of the eukaryotic cells, and then this mature mRNA is going to come out in the cytoplasm, and it will act as a template to produce proteins through the machinery of translation. So now we will look into some previous year questions of the NEET exam. So let us see the first question. What is the role of RNA polymerase 3 in the process of transcription in eukaryotes? So as we know, the RNA polymerase 3 is going to help to produce tRNA. So the option number 4, which says transcribes tRNA, 5S RNA and the snRNA. So this option will be the correct option. This question has come in NEET 2021. Coming to the next question, which is the only enzyme that has the capability to catalyze initiation, elongation and termination in the process of transcription in prokaryotes? So the only enzyme is DNA dependent RNA polymerase. So this question is also coming in NEET 2021. So this type of questions are going to be asked in the NEET exam. I wish all happy learning. Thank you.